All right, so our last screencast, we were looking at the first step of cellular respiration, which is glycolysis. And little quick status report at this point, glucose has been partially broken down to two molecules of pyruvate. Um, we harnessed some of that potential energy from glucose as ATP, and we've also harnessed some of it in this form, this kind of intermediate form of NADH. And but now we can continue to break pyruvate down even more because remember in cellular respiration we break glucose down to carbon dioxide. So we're not quite to carbon dioxide yet. So let us continue our journey and continue to break down pyruvate. So the next step in cellular respiration is pyruvate oxidation. Sometimes this is also called the transition reaction. And this occurs in the mitochondrial matrix. And so here you can see that after glycolysis, that pyruvate is going to be transported into the mitochondria, and this transition reaction called pyruvate oxidation will take place. And the net reaction is that pyruvate is converted into acetyl coenzyme A, which we see right there. And in the process, a molecule of CO2 comes out, and a molecule of NADH is generated, and that's shown in this diagram as well. So these are kind of showing the same thing. So let's break that down. Here it is again, and if we look at the actual numbers, we see that we get 2 NADH specifically from pyruvate oxidation. We get no ATP from substrate level phosphorylation, and then again, we're going to ignore this third column for the time being because something weird goes on later. Okay, so, oh, and actually, sorry, one last little piece. This CO2 that gets broken down, where does that CO2 end up going? Well, where it ends up going is into your bloodstream, where it is ultimately exhaled by the lungs. So every time you go and you're exhaling, you are actually getting rid of the CO2 produced by this process. The other thing I wanted to point out is, remember, this is happening twice per molecule of glucose. So again, glucose makes 2 molecules of pyruvate. You might want to add that to your figure. And then that goes in, and those 2 molecules of pyruvate give rise to 2 molecules of acetyl-CoA, giving off 2 molecules of CO2 and 2 molecules of NADH. So let's continue then. The acetyl coenzyme A that is produced as a result of this then goes on to what is called the citric acid cycle. This also occurs in the mitochondrial matrix. And what you see is that acetyl-CoA enters this little circle thing, and then we get kind of a lot of stuff out of this. We get NADH, another electron carrier called FADH2. We get ATP out of the deal. Sometimes there's actually a place where you get something called GTP, which is basically the same thing as ATP. It's just an energy source. And then you get more CO2 out of the deal. And so you can see that too. There's the CO2, there's the ATP, the FADH2, the NADH. And if we wanted to quantify those numbers, oh, we have arrows. There's the arrows, in case you missed that. If you want to quantify those numbers, here they are, eventually. Arrows again. There it is. Okay. So we end up getting 6 NADH, 2 FADH2s, and 2 ATP. So just like with glycolysis, we're going to want to take a look at how we account for these numbers. And so for that, we need to look at another big, scary diagram, this biochemical pathway that shows all of this. So before we get into the pathway, I just want to point out that the citric acid cycle also has several other names that it goes by, because in biology, why have one term when you could have 1,000? So the citric acid cycle is also called the Krebs cycle. That's named after the scientist who helped kind of figure this out, Dr. Cycle. Just kidding, Krebs. It's also sometimes called the tricarboxylic acid cycle or the TCA cycle. The reason for all these names is you'll notice that this reaction here is that pyruvate oxidation stage where pyruvate is converted to acetyl-CoA. When that acetyl-CoA comes in, it forms citrate. So that's why it's called the citric acid cycle. And you'll notice that citrate has 3 functional groups, which are carboxylic acids. that C double O, double bonded O, and then that O, that's a carboxylic acid functional group. So that's why it's called the tricarboxylic acid cycle as well. So that's the names kind of make sense based on the immediate starting point of that Krebs cycle. So let's break it down. So again, 2 carbons are going to enter the Krebs cycle as acetyl coenzyme A. Okay, so this acetyl-CoA is a result of pyruvate oxidation, and so it's going to enter. So we have this molecule, you don't need to know the name of this thing, it's called oxaloacetate, but you'll notice that it's 1, 2, 3, 4 carbons long. We're going to add acetyl-CoA, which is 2 carbons, and we form this 1, 2, 3, 4, 5, 6 carbon citrate. So 4 carbons plus 2 carbons yields 6 carbons. So 2 carbons are being added to this as acetyl coenzyme A, and then down here, 2 carbons are actually exiting as carbon dioxide. So, and just like with pyruvate oxidation, those CO2 molecules end up being exhaled. So the only reason why you need to exhale is to get rid of the CO2 produced by this process and pyruvate oxidation. That's it. That's why you're exhaling. So basically 2 carbons in, 2 carbons out. This is part of why it's a cycle, right? Because we get 2 carbons in, 2 carbons out. We also start by adding those 2 carbons to oxaloacetate, and then at the end, we regenerate oxaloacetate. So again, it's a cycle. 2 carbons in, 2 carbons out, regenerate oxaloacetate. 2 carbons in, 2 carbons out, regenerate oxaloacetate. Cycle. All right. Along the way, a number of chemical reactions are taking place, each of which again has a negative delta G or is coupled to an exergonic reaction, and each of which has its own enzyme, which is listed here. We have one substrate level phosphorylation event where this molecule takes a phosphate group and then this enzyme adds it to ATP. And then we get a lot of these electron carriers. So we get one here, one there, one there, one there. Again, NADH and FADH2, very similar functions. Now, if you look at this chart here, I counted 3 NADH and 1 FADH2, but here it's saying 6 and 2. What's up with that? And hopefully you remember everything is happening twice. Since each glucose gives you 2 molecules of pyruvate, we have to multiply all of this by 2. So 2 pyruvate, and you can add this to your sheet, gives 2 acetyl-CoA, releasing 2 CO2 and 2 NADH. And so then we get 2 NADH here, 2 NADH here, 2 NADH there, and then 2 FADH2. We get 2 ATP down here. And so everything's happening twice per molecule of glucose, and that's why these numbers are 6 and 2. So basically the Krebs cycle turns twice per molecule of glucose. So you'll notice also by the time we're done that since we lost 2 CO2 here and 2 CO2 here and 2 CO2 there, that accounts for the 6 carbon dioxide molecules that are released. So by the time we're done with the Krebs cycle, we have completely broken glucose down into 6 molecules of carbon dioxide. And we've harnessed that energy as, I'll go to the next page here, 4 molecules of ATP and a beep ton of these electron carriers. So we've converted the chemical energy of glucose into some other things, and we have a little bit ATP, but in our final step, we are going to have to deal with these electron carriers and use them to actually make a lot more ATP. And so that is what our next screencast will cover.